maybe not. Hey, everybody. Thank you so much for making time to join us here at the Global Genes Live, a rare patient advocacy unsummit. We're really, really excited for this session. This is this panel. This is something I've had so much fun talking to everybody in prep and really diving into this, and I'm excited to lead this panel. So thanks so much to Global Genes for having me. My name is Luke Rosen, and I'm the co-founder and board chair of KIF1A.org. I'm going to be your moderator today, so it's my great honor. Before we kick everything off, I just want to go over a couple of important notes here. First, all of the attendees are currently muted by the moderator to reduce any interruptions or, if you're like me, screaming children in the back and other things like that. We encourage you to use the chat function on the On2 window for any questions that you might have during the session. And after, when it comes time to Q&A, the moderator team is going to kind of present the questions, and I'll relay them over to our panelists. To open the chat, just so everybody knows, you go down to the blue chat button located at the meeting controls at the bottom of your screen. If there are any questions and you're not able to get them addressed by the speakers, then we're going to do our best to try and catch up and answer them later on. And don't forget, guys, to post your top takeaways and fun moments during the Global Genes Live on social media. So it's at Global Genes, and you're going to get featured on the social media channels, hopefully. And the hashtag is #2020GG Summit. So finally, we are encouraging everybody to participate as active listeners and take great advantage of this opportunity to hear from these three really remarkable leaders and experts, and ask them questions. We're going to try to just have a lot of dialogue here. So now I'm pleased to welcome to our discussion, the program. This is your time to find answers to challenges and things that we're all talking about here. So I'm really pleased to welcome to this discussion Hetanshi Naik, who is the assistant professor at the Icahn School of Medicine at Mount Sinai. We also have Nathan Peck, who's the CEO at Cure VCP, and Megan O'Boyle, who is the head of engagement at RareX. Each of these wonderful leaders is going to share a more complete background as we jump right in. So we do have polls going up. So the first poll should be there. Megan, do you see the poll?
It's up. It's up.
It's up. Well, thank you. which category best defines your organization? Thank you, Molly. I'm having trouble seeing the poll, so thanks for letting me know. So this is the first poll question, is which category best defines your organization? And this is a really good one because something that's, I think, really useful about this panel and what's so special is that we have three leaders who have natural history studies at different stages along the journey. So I think it's really important for those of you out there who can weigh in and see where you are and ask questions of the folks that are a little bit either further along or right in your wheelhouse of the journey. So we're going to leave the poll up just for another second, and then we're going to go to the answers. And make sure that you scroll down to submit your answers. Thanks for bearing with me, everybody. So let's see, patient advocate in early stage is 24% of our audience, which is great. And patient advocates with pre-drug experience is 12% of our audience. And then patient advocates with drugs in pipeline is 30% of our advocates. And then we have 12% of our audience are researchers. So that's really fantastic. Thank you guys for answering that poll and engaging where we all are in this natural history journey. So now, it's my great pleasure just to get out of the way and invite Hetanshi up to talk about.
Too early. I think there's one more question before we get to me.
Okay, thank you. Sorry, we have one more poll question, and Megan would have killed me if I skipped right over it. So thank you. The next poll question is, does your disease have a natural history study? So this is a really important, obviously, and relevant poll question. So thanks for going back. So same thing, if those in the audience could take a moment and just hit that poll, it's really going to inform the conversation and drive things along. So again, the question is, does your disease have a natural history study? Thanks, Molly at Global Genes, for relaying these poll questions to me. I appreciate it. So 42% of our audience says, yes, there is a natural history study happening, and 57 saying, no, there's not a natural history study happening. So this is going to be a really useful and fantastic panel for, I think, everybody. So with that, now I will invite our wonderful guest, Hetanshi, up to present.
Thank you. So I'm just going to give everyone a little bit of background about myself. I am a genetic counselor by training, and I also have a PhD in clinical research, and I've been doing clinical research for 10 years. So I primarily work with the porphyrias and the lysosomal storage disorders, and I'm an investigator with the Porphyrias Consortium, which is part of the NIH-funded Rare Diseases Clinical Research Network. So I have been involved in all kinds of different studies, from designing intense longitudinal studies to simple chart reviews, cross-sectional studies, kind of the whole gamut. So I'm really happy to be here today and hope I can share my experience and it's helpful for everyone.
Is that RDL, this version of this RDL?
Now can I invite Nathan up to introduce yourself?
Sure, and I had some slides. So Nathan, am I going into my slides first, or? Okay, Nathan Peck from Cure VCP Disease. And again, we saw the diversity in the responses from the audience. And so, I wanted to make sure, that I introduce our organization and myself. I have 5 minutes. I'm from the South, but I'm going to talk quick. So, again, it's important to have that lens, of what stage your organization is in. And so I briefly wanted to introduce that. So, I am a patient with VCP disease. I was diagnosed about 5 years ago. And VCP is the valicin-containing protein, a P97 gene, also called IBM-PFD or multisystem proteinopathy. It's an adult onset disease, and it's autosomal dominant. We don't know how many families have it because so many patients get misdiagnosed with the disease. So commonly misdiagnosed with limb-girdle muscular dystrophy, sporadic IBM, myositis, multiple sclerosis. And so the challenge with our disease is that it has multiple disparate phenotypes. So most patients will get inclusion body myopathy, which my mom is deceased from the disease, my aunt is deceased from the disease, and I have 2 uncles with the disease. My mom had inclusion body myopathy and Paget's disease of bone. But we have, and so a patient can get 1, 2, 3 of those phenotypes, which affect multiple, disparate systems. We also have patients that have ALS as a phenotype. And in very rare cases, there's Parkinson's as well as Charcot-Marie-Tooth. So the challenge from our standpoint is, there's a lot of different systems affected. Within families, you can have different, phenotypes. And so 2 years ago, I'm honored to be a part of this because 2 years ago was my first Global Genes. And so I never, Luke was actually getting the Rare Champion of Hope Award in 2018. So it's pretty awesome. And I'm very honored to be here with everybody. But we started our organization in 2018. We're the only patient advocacy organization for VCP and IBM PFD. We're entirely volunteer-led. We have a 3-person medical advisory board. And our primary goal is patient identification. We have to find the families because right now we have no natural history, biomarkers, or endpoints. But want to speak to you about what we're doing and hopefully offer you some practical solutions. So, I'm an engineer, I'm retired on long-term disability, but our primary focus right now when we're speaking of natural history study is we need to understand the rate of degradation of our disease. And so I wanted to share this quote from Lord Kelvin back in 1883 that I often say when you can measure what you are speaking about and express it in numbers, you know something about it. But when you cannot measure it, when you cannot express it in numbers, your knowledge is of a meager and unsatisfactory kind. And so that's kind of the principle that, how we're approaching, we have to go get data about our disease. And so how we did that in the beginning was a patient registry. We started a contact, but also patient-reported outcome registry. And we started that 5 months after we started. It's hosted with Cords originally, and we just revised it this month based on learnings from researchers that had already tapped into the data. And so it started at 69 questions. It's now at 87. We pulled all our questions from healthmeasures.net, which, has functional rating or has already standard questions that are scored and validated and also offered in different languages so that hopefully we can offer our registry in different languages and just use those already written questions. We also have some functional rating scales in there, and we ask demographic questions. The exciting news for us is as of Sunday at Washington University in St. Louis, they did a report or a publication on our registry, and that actually is going to be published in the Orphanet Journal of Rare Diseases. So we're very excited about that in terms of broadcasting the message, out to the research community about our registry. And we started with 5 sections, and now it's 6. And so the message there is you start with something. You're always, and I know Megan, she's had this challenge as well. There's always going to be learnings, but for us, it was so important to get something out there to start learning about our patient population. So when we're talking about natural history study, we need a longitudinal functional measure study. So the patient registry is, patient-reported outcomes, but we actually need to measure. And so, we want our study, the functional measure study, to be agnostic, not tied to one research institution. Like all of us, time is a force against us. Gravity is a force against me, as I'll be in a wheelchair in another couple of years. But, we don't want to wait, for somebody to have to write up a study. Budget, we've got limited funds. We know natural history studies are expensive. But what's happened, and COVID's really opened up an opportunity is there's one institution that's been working on the neuromuscular community in terms of these functional measures. And so we want to try to leverage that organization or that institution And the question for us was, does COVID present an opportunity to execute a remote study via Zoom, for our patient population where we don't have to create these centers of excellence? The question we get asked a lot of time, there are some repurposing opportunities out there. The first question we get asked is, what does your natural history data look like? And so it is imperative upon us to go get that information. And so just some practical, things that we're doing, we're trying, learning, and iterate. We don't want to sit around and wait. And so I just wanted to share one example or two examples of what we're doing. We bought dynamometers because hand grip weakness is something that, over time you can measure. And so we bought 100 of these dynamometers. Our goal is to present them to the patient population. We started with our board and some healthy individuals. We set up a Google form. All the data goes in the Google sheet, and every 2 weeks, using Zapier and text magic, we send a text message to our patient to all the participants to remind them. And so every 2 weeks, we're getting data. Now, the challenge is, we had to switch. Well, I switched over to G Suite so that we could use, a BAA to have a HIPAA compliance. We need an IRB, set up for this kind of homegrown study in order to publish that research. But in the meantime, we're just starting because we feel like this is a great opportunity to condition our patient population for how to measure themselves and use tools, to start getting data on the rate of degradation. And then the other exciting thing that we're doing is this remote functional measure study. So we talked about that one institution. It's going to be led by one PI. We've selected validated tests, which I have on the next slide. But we're first starting with a proof of concept, which is 5 patients over Zoom, just learning what challenges we might run into. Phase 2 is validating these remote measures that there's no difference between the clinic and Zoom. That's something we have to prove to the scientific population. But what we've done is we bought kits, for each of these patients that, tripods to put their cameras on, little step stools, spirometers that people can measure the FEV values. So, and there's a lot more equipment that is in there, but we're basically trying to replicate the clinic versus home so that this opens up the opportunity to engage more patients, to do it in a much faster time frame. And so we're very excited about that. And I want to thank, and we're also looking at data systems that we can get everybody's medical records and everything into one system. And so it's in the preliminary stages, but we have started phase 1 already. And I did want to thank Global Genes. We had to pivot on our rare impact grant, and they allowed us to do that to put this kit and equipment together for our patients. But again, I want to make sure we have enough time to, for the discussion and Q&A, and I'd be happy to answer any more questions that anybody has about that. Thank you.
Thanks, Nathan. That's pretty incredible. And, we have some questions coming in already, and I think we're going to get to those questions. But first, I do want to introduce Megan so we can all have a kind of a rich dialogue together. But I think it's important to just kind of clarify a few terms, right? Because a lot of times they get amalgamated together. So between, things like registry, natural history study, and longitudinal study, right? And so I think that one of the, there are differences, and I know that our incredible panelists will kind of dive into them. But just, Nathan, after hearing first of all, congratulations and well done on all of this. It's a Herculean task what you're doing, certainly in such a short time. But one thing that you mentioned resonates certainly with me and our organization and our KIF1A families, but the heterogeneous nature of the disease and how important it is to somehow capture the variability and the challenges that everybody faces. And I think that, I'm sure that Hetanshi and Megan are going to be able to speak to that too. So maybe something just to keep in mind. And the second thought that I had that you brought up is one I know Megan is going to speak to a little bit is how can we kind of capture the longitudinal element and the longitudinal data in disease and address it sort of rapidly, right? Because we don't want it to go too long. So with those two thoughts and takeaways that I had from Nathan, thanks. I'm going to bring up and pass it over to Megan O'Boyle, our good friend and leader. Thanks, Megan.
Thanks, Luke. And my favorite part is Q&A from everyone else. So I'm going to move quickly, and it's hard not to be able to see the audience. So hello to all those who I've seen, and my apologies if you've heard these words, and a special hello to the people that are new to Global Genes. So my main title is Shannon's Mom. I've had that for 20 years, and 19 years ago, Shannon was diagnosed with Phelan-McDermid syndrome, called 22q13.31 syndrome. And as you know, a lot of these things get renamed. I'll be referring to it as PMS to make things faster. And there was very little known about that, and very few, less than 50 patients in 19 years ago. 2,400 patients known in the world. As with every rare disease, it's underdiagnosed, and especially underdiagnosed in underrepresented communities. About 10 years ago, our foundation that was started by parents like Luke, who had the courage and the passion to start a foundation, reached out to me to help with a registry, and I didn't know what one was, and so I had to learn. And with the help of a bunch of other parents, we navigated how to start a registry and launched it, and asked 300 questions over the last 10 years. It was meant to be a longitudinal study. It was meant to be check-in every time you go to the doctor, or at least once a year on your child's birthday. But based on the way the questions were asked, and without skip mechanisms and so on, it was too much. It was a burden. So we do have almost 70% of our worldwide family population enrolled in the registry, but we have not been able to get, annual touch points as we had hoped. Thanks to funding from several national institutes of health, we have under the NCATS, it's called Rare Disease Clinical Research Network, RDCRN. Every 5 years, there are 20 so consortia that are awarded this 5-year award from multiple NIHs, and there are at least 3 diseases per consortia. And we are fortunate to be heading into our second round, which means our 6th year. This does include a more traditional natural history study in which patients are seen in clinic over a period of time. Over the 5 years, our patients are seen 3 times, so not quite every year. There is a protocol that all the sites, so we have 6 different places around the United States, have to follow. And research and publications come from that. That data from RDCRN is shared with outside researchers, which is great. Just recently, I became involved with something called RareX. If you're not familiar with it, it's rare-x.org. And this will be a global, it is a nonprofit, and will be a global platform that will collect data as registries do for rare diseases, any rare disease. And it will also be a platform for data access and analysis and distribution. So there will be a federated data platform where RareX can connect to existing data sets like Nathan's, or like the RDCRN data, for instance, or clinical trial data, or biosample data, or wearables, or apps. It is to take advantage of current data as well as help disease groups collect data. So that was a little longer than I wanted it to be. Two of the things that I think we're going to really touch on today is the barriers to natural history studies, and also how we as patients and advocates can actually help with natural history studies. And I'm looking forward to that conversation. Going on mute.
Awesome. Thanks so much, Megan. I think, two questions that came up, I think you kind of touched on a little bit there. And so I know you just went on mute, but I might kick this one right back to you. One main question is, how and where do you get the questions that you integrate into your natural history study, right? And I think that that's a really important question. The other part of that, I think, is how do you consent your families to actually answer those questions? And I think maybe Megan and Hetanshi, you can speak a little bit to it too from the clinical end. So maybe Megan, do you want to briefly talk about how you get the questions that you end up putting into your study?
So in 2010, we went to researchers and like organizations, so organizations that had diseases that looked like ours: seizures, autism, intellectual disability, difficulty perspiring, and we borrowed their questions. But we didn't know enough about informatics and how that data came out on the other end. And when our data started being analyzed, we found that some of these questions did not yield the data that we had hoped for, or that industry, researchers, and most importantly, FDA would find valuable. I've learned a lot since then, and there are lots of standardized measurements, just like what Nathan spoke about when he said that they use the health measures. The National Institutes of Health has a tremendous library of medicine full of standardized measures. And by that, it means that you are asking the question the same way, and it's been validated in some way. I will caution you not to be Debbie Downer, but just because something's called a common data element and it's been a vetted, standardized measure doesn't necessarily mean it's going to yield the data that FDA and industry need or want. So I would say less questions is better, but make sure that what you're asking is standardized. Do not have fill in the blank, I beg of you. And make sure, if you can afford to, work with informaticists, statisticians, survey methodologists. There's a whole group of scientists out there that I don't know. But this is what RareX is, part of RareX's mission, is to make sure that everybody that has a certain symptom, no matter what your rare disease, is asked the same question. So if this is about muscle degeneration, any disease with muscle degeneration would ask the same question, so we could do cross-disease research. Sorry, long answer?
No, that was great. You were going to say a lot of things.
I want to take one step back, and Hetanshi, also kick this one over to you. I think we need to step back and actually, I know the slide is up there, so thank you for putting the slide up. What is a natural history study? And maybe Hetanshi, if you don't mind just touching on that, and then speaking to the clinical elements of it, that would be great.
So the traditional definition of a natural history study is the one that I have up here on this slide, which is a pre-planned observational study that is meant to track basically disease outcomes. And that can be demographic information, genetic information, environmental factors, disease comorbidities. It's very broad in nature. And how that compares with a registry study is the registry is a very, very broad term, and natural history studies can be registry studies. So they are often used interchangeably, even though there are some nuances. So generally, a registry is just a broad term for some way of collecting standardized information. And it usually is more broad in scope than a natural history study, which can have a defined outcome. So I'm just going to put up another slide just of terms. I'm not going to say these out loud, but just so that everyone has them. But in answer to kind of the question about where do you come up with the questions, especially as researchers, the first thing we do is you go to the literature, you go to, you see what's being published before, what are patients asking about, you want to get.
I'm so sorry to interrupt you, but I'm going to just remind everybody to mute. If you're not on mute, we're hearing some folks in the background, and I really do want to hear what Hetanshi has to say here.
Yeah, I apologize. That might, in fact, be my background because I have someone doing their own conference call over there. So I will just try to be louder. So the place that we go to for the questions. So as a researcher, the first thing that I'm going to do when I see something that either patients are asking about something, you go to the literature, it's not in the literature. So you want to think about how to define it and how to capture that information. Engaging patients and patient groups is very, very important in hearing, what their experience has been. And that's how you can figure out, one, is there some existing set of scales or something that I can pull and use that? Or is there truly, you've looked at something, you've talked to a bunch of researchers who see patients, you've talked to a bunch of patients and got their opinions on some of these existing scales, and it's not something that seems to fit quite right. And that's when you start to think about, okay, do we need to design something new, which I do say with some hesitancy, because designing something new is an involved process. So, as Megan said, you want to make sure you talk to the right people, you get the right expertise in terms of statisticians and survey development experts before going down that road. But I do think it's important to say there might not be anything out there that fits quite right.
And I think that's a huge point, right? Especially in rare disease, is that, and now speaking purely from my own opinion from our foundation, KIF1A.org, is that, there really aren't appropriate measures for what our challenges are. And what I think our responsibility is to really mobilize and create those new measures if we really want to be able to change what matters most, or to be able to capture that change that matters most. And I wonder if there are two utilities for a natural history study. And I'd love to hear each of you kind of respond to this. And I might be way off, but I see the utility of natural histories being twofold, right? One is more long-term and one is more near-term. The natural history study in the longitudinal sense, being able to have a picture of disease and the sort of the life cycle of the disease. And then there's a more tactical use for a natural history study. And that could possibly be to take the place of a placebo arm during clinical trial. Because if it's so valid, can it be that control? So I don't know, maybe Nathan, do you have any thoughts about either of those two?
Well, I mean, we have a small patient population, so and what everybody else talked about. I mean, we're defining what we're trying to do, and we know some of it's probably going to be wrong, but as I said in one of the slides, we have to understand the rate of degradation. We have to be able to quantify that. If we're going to do a clinical trial, we need to know if something's working. But yeah, we also are fully prepared that we might have to use our patients as their own control. We haven't gotten that far yet because we need to have the baseline data first of all. So I don't know if that answers the question, but it is a challenge, certainly.
Yeah, it does. And Hetanshi, what do you think about?
Before Megan, I just want to make sure that everyone kind of is thinking about this, and that longitudinal can mean very different things. It depends on the disease. So longitudinal could be a 2-month study where you're assessing something maybe weekly because that's how quickly things change, or it could be several years because that's how the progression goes for the disease. So I just want to make sure that point is out there before punting it to you, Megan.
Yeah, Megan, go ahead. I think you have a point that you wanted to bring up that is relevant. And I know people are asking about data ownership, and I'm going to say I think that's a separate conversation, but if you want to touch on it, please do.
Several things. One, I want to back up and say, when I said have the right science around the informatics, I don't mean each and every individual group. I mean, if there are not standardized measures that your research community or industry partners or what you think FDA would think are good value measures, and you have to come up with new ones, as was mentioned before, you're not going to do this by yourself, and it shouldn't be at your expense. There is, as rare as the 9,000 plus diseases are, we all have a lot in common. I can tell you my daughter's syndrome looks like identical to 50 other syndromes with the exception of lymphedema and weird toenails. That means I should not be making up a PMS assessment specific to us. I should be borrowing Angelman's or Rhett's, another organization where there are non-speaking children who are poorly assessed with current assessments. I beg of you guys, do not one-off this. That is where we are trying to change rare disease, because then we can't do cross-disease research. So for those who do not have what's called a registry, please look at rarex.org. We are only in the pilot stage. We are not ready to start. So if you have to start tomorrow, do it. I'll give you some advice on what you need to know. All patients own the data. But the other thing I want to tell you is work with organizations like yours. If you are an autoimmune disease, look to see what questions the bigger groups are using. If there's a need to come up with new standards, work with all the autoimmune diseases that need the same standards. This should not be something anybody does one disease at a time.
So this is a great point, and I think that, this answer leads into one of the questions. And look, Megan, our daughters have very similar challenges, and I imagine that in our natural history studies, there's overlap, right? And so I'm wondering where can, if there's a foundation just starting out, with their natural history study, is there a place, or is there, I know, at least from our foundation, we're happy to share our questions with everybody. But I'm wondering, is there a place where questions live that people who are just starting out can kind of pull from a natural history study? And if there's not, should there be?
If they're neuro questions, the first place to go is the National Institute of Neuro and Stroke, NINDS. And it may be referred to as common data elements or standardized elements. If you're looking at cancer questions, go to the National Cancer Institute. If your eye questions, go to the Eye Institute. But I also, if you take something off the shelf, make sure you have some feedback from your researchers. And if you are fortunate, God bless you, if you're fortunate enough to be working with industry or in the pipeline, then your industry partner should absolutely be giving you the stamp of approval. The problem with standardized questions is there are too many of them, and it becomes very overwhelming. And I'm sure Nathan can tell you, when he went to look for demographic questions, there were tons of standardized questions. So don't get overwhelmed. Something's better than nothing. You can always fix it later. The most important thing to Luke's question about 10 minutes ago was consenting. Do not wing it. You need governance that will get you through this process and not bite you later. In other words, make sure you know how to consent your families to share de-identified data. And I strongly encourage everybody on this call who is all have a stake in rare disease. We should consider families in natural history studies, whether they're government run, institution run, university run, PI run, to say we want patients to have the option, not the mandate, to share identified data safely with sources. This is a very disruptive concept, but I think it needs to come from the patient community. And so, Megan, we can have a whole other day.
Yeah, no, I think this is a really important point. And I'll just say, and then I'm going to ask Nathan to talk about what sort of tactics you might be using to accomplish this. I think that natural history studies are a lot of times are done wrong, right? And I hate to say that, but why they're done incorrectly is there's really one goal that we all have, right? And that goal is to develop treatment as fast as possible for our patients and for our kids and for our loved ones, right? And so to me, that means that a natural history study needs to be powered for translation, right? And so I think that how you do that is by asking, right? Is just by going and asking, and you can ask for guidance from FDA, you can ask for guidance from industry partners. I certainly, I work in biotech, and we daily have different fellow advocates and foundations calling in and saying, hey, can you help us in figuring this out? So Nathan, I'm wondering if you and your group are doing anything to proactively engage stakeholders like industry, like regulatory agencies, and integrate their sort of feedback into your natural history study.
Yeah, it's a great question. I mean, the first stakeholders we talked to were the researchers and scientists, who want this information, but whether it's priorities or other projects, they just haven't done it. And so we have had guidance from them on whether it's the questions, the patient-reported outcome questions, or even the functional measures. They've recommended that. We did our first patient listening session with the FDA back in May. So that was kind of our first foray into even introducing the disease to them. They didn't know a whole lot about our disease until we talked to them. So certainly one of our goals is a PFDD meeting at some point, but again, we've got to have more data and something to present to them. So, the drug company, we've talked to 3 different, drug companies. And again, they're very interested in us, but they need some data behind it. Now, have they given us specific feedback on what they want? No, other than, that rate of degradation. I keep bringing that up. They want to understand, because our disease looks a lot like ALS, but just on a much longer time frame. So, from me 2 years ago to now, yes, I have gotten a lot worse, but what is that rate? And so, that's pretty much what we've been asked to provide. So, again, not a great answer to your question, Luke, because we're just so early in this.
Right. That's. Thank you, Nathan. I think that that's really important, right? And that to Megan's point, that makes that easier, that engagement with different stakeholders, is the difference between de-identifiable and recontactable, right? Because you need to be able to ask questions of the community if we're really trying to figure out a way to develop those treatments. And before I pass over to Hetanshi to talk more about how clinicians and how taking those data actually can help on the ground. I'm going to directly. So we've been kind of taking pieces from questions that are coming in, but I'm going to just verbatim read one question. Right.
I'm going to interrupt first. Sorry. When you say translation, for the 24% of the early groups, translation does not mean from English to another language. In the way that Luke's talking about, it means from, animal model studies and test tubes and petri dishes to drugs in your capital. That's, if there's anything that we've confused you on, please feel free to put it in the chat or send us an email. Sorry about that.
Thank you for clarifying that. Yeah, when I say translation, I mean from preclinical research into clinical development of therapeutics, is translating that research into actual treatment. So thanks, Megan, for clarifying that. I'm going to let's see. I'm going to ask for an here's a question that came in. For an ultra rare disease with less than 5 children and young adults alive at this time, under 200 total known to be diagnosed, how would we create a path to begin a study? I'm going to actually kick that question over to Hetanshi, if you don't mind answering how an ultra rare community might go about doing a study.
It's definitely tricky, but it actually goes back to what Megan was saying, which is try to find something that is similar, and it might not. There might be still a lot of differences, but if there are groups that are similar enough that you can partner with, it might be a start in terms of just getting some support to try and get a registry going or collect some early data points. And the other thing that I just want to touch on briefly, especially for some things that are ultra rare, is the benefit of qualitative studies. We're talking a lot about quantitative studies, which is you're getting data points, you're getting things that you can analyze in a very specific way, you're going to get numbers. But for some things that are ultra rare, as you're saying, five patients around the world, hearing those patient experiences and putting it into a qualitative study, which is still done in a systematic way, can be very powerful and very useful. So it might be a place to start where, doing a focus group or an interview study, something along that line can still give very useful information for the community.
Yeah, that's a great point, the difference between qualitative and quantitative. And I will add to that is that, and this is just my sort of, something that I see as a success is, for multiple communities, but I'll use ours, is that if you're just starting out and if you were to make one priority or one job, right, of yours as an advocate, as a foundation leader, I would say that it would be to engage your community and activate them in a way that you have rapid response from a high percentage of your community. So I'll give a quick example is that in our KIF-1A natural history study that we have running right now, we needed to drop a survey into the community to add a data point about seizures. And we worked with our academic collaborators to put that survey into the community. And we had 97% response rate within a week. So I think that the success doesn't come from the actual questions. I mean, they will, but that's really the clinician's job, right? I think that the success from an advocacy point of view is to be able to engage your community and make sure that everybody who is willing and who is able is participating in this study. So that's my one bit of advice for a new group just starting out is do whatever you can to engage your community and make sure that even if you have a disease with 20 people diagnosed, make sure that all 20 are involved. If you have a disease with 200, same thing. So that's my bit of advice. We have just a few minutes left. So I want to go around and just to ask each of our wonderful panelists if they have one bit of advice for a new group starting out. Maybe we start with Hetanshi.
Sure. The thing that I have seen to be the biggest issue in studies is their design is that they don't have a clear goal. You know, sometimes it's designed with like, we're just going to catch all, we're going to get as much data as possible, but in practicality, how that works out isn't quite as well, you know, the data that you end up getting isn't that useful sometimes. So if someone is just starting out and you're thinking, you know, what can we do? I would say just try to, it's okay to start small and then try to define the goal of the study clearly at the onset. And it's okay to have a broad goal, but then just make sure as you're designing those specific questions or data elements that you're capturing, it's clear what you want to do with those at the end.
Great. Thank you. That's such important advice. Nathan, you want to give us a quick couple of words of wisdoms for foundations just starting out?
Yeah, I mean, just try, iterate, and learn. I mean, what we've kind of found is one of our principal investigators, that discovered the disease, has been sitting on her study to be published for 10 years. And because we've done all this activity, and it may have been not the right stuff, but it's got her motivated and moving now, to where she's going to publish. And so I think, just as I showed earlier example, putting something out there, giving, our patient population now has a little bit of hope because we're measuring some things, where there was nothing before. So it just shows some activities back to that engagement thing that you talked about, Luke, which is really important, getting people more engaged.
Yeah, that's great advice. Thanks, Nathan. And over to you, Megan, for last words of wisdom here. Oh, you're muted, Megan. So I'll ask a question. I'm new, just starting out. Do you have advice for me?
Yes. If you need advice on how to start a data collection as in a registry, there's an NCATS drug development toolkit and something called Radar at NCATS. And I've put my email in the chat, and I'm happy to direct people to those existing resources. From a natural history study, the traditional natural history study, if you're fortunate enough to get into clinics where clinician researchers are doing protocols, that doesn't mean that we just show up as patients and we give our information. It means that your patient organization, prior to that protocol being finalized, should ask to see the protocol and give your feedback and say, I don't think our kids can handle that in 4 hours. Can you make that a longer visit? Or I think I think the EEG should be done before lunch instead of after lunch. To that point, you should also ask to see the consent. If it's dozens of pages, it will not be an informed consent, because if you have a child like mine and you're standing in a clinic and you have to review dozens of pages, you're going to skip to the last page and sign it. And that's not informed. It's what we call an iTunes consent. You scroll to the bottom and you sign. So patient organizations should be asking to see these things and helping these researchers put things in ways that families can understand it, in shorter versions, less repetitive, and a little less scary. I find that sometimes these consents are protecting us from things that are never likely going to happen. And also, these organizations can help with recruitment. I think researchers in industry are so conditioned to separate themselves from patients that they forget that we can actually help them. And I'll stop there.
No, that's a really great point. And just to wrap it up quickly, is that I think the natural history study and our duty in creating them is to say what matters most to the families in our community and create a picture of how we can change that and have a meaningful therapeutic change. And so just with that, I want to thank our panelists, Nathan, Hetanshi, and Megan. You guys are just incredible, and I'm happy to know you and call you colleagues. And to all of our audience members, thank you so much for joining. And make sure you stay tuned here for the next session, which is proactive planning for continuity of research in crisis. Wonderful job, everybody, and thank you guys so much for joining us. Sending my very best to you and your families.
Thank you.
Thank you. Hashtag 2020 GG Summit. Have a great day.
Bye.
Bye.
Thank you, guys. That was really wonderful. Yeah. Make some cash.
Wait, were you like the only person on this meeting?
No, I was actually moderating a meeting.
What was that?